after the exam, I'll probably catch up and get ahead of it. All right, so our last clade that we have to go through is the amorphia. Okay, you'll also see it called the uniconda, but in here we're referring to it as the amorphia. And it consists of a group clade called Amoebozoa, fungi, and animals. But on this particular slide, or this particular lecture, topic 6, and this will only be Amoebozoa and the fungi. We have another lecture 7, that's going to be the animals. Okay? All right, so going with these, this is sort of the breakdown of this particular so-called supergroup, is that, of course, I already mentioned before, apomorphies for the clade is the triple pyrimidine fusion gene. Okay, that's a gene that was 3 different genes that have fused together in this particular lineage. Okay, so it has one transcript that when it's translated, it will be divided into these 3 different enzymes. All right, that's unique because in all other groups of eukaryotes, those 3 genes are separate from each other. They have a separate promoter. They are transcribed separately. Okay, and then the reduction of the flagellum to, when it's present, to just one flagellum. All right, and so one clade here is the Amoebozoa, and then another clade, such a clade, involves fungi and animals, and this clade here is called Pistoconda. I'll give that up in a moment. So single flagellum, that's these down here. Go back here. I'll come to that word in a moment. This clade has the name, it's called Pistoconda. In fact, let's go ahead and write it up on the board. It's going to be up on the slide momentarily. And the apomorphy for that is that the flagellum is posterior. Down here in the Amoebozoa, when there are flagella, it is an anterior flagellum. Now, how can you possibly tell whether a flagellum is posterior or anterior? By the direction in which the cell happens to be moving. Okay, so in animals, for example, sperm cells, the flagellum is posterior. Okay, whereas in this group, when they have flagella, it's anterior. Okay, so look at this one clade called Amoebozoa. I'm not going to spend too much time on it, just only mentioning some of the things that happen to be here. These are lobate amoebae and slime molds. You don't hear too much about slime molds, but they're rather cute. And some of them are multicellular masses like this up here that are moving like amoeboid motion, but there's no cell divisions in there. It's just one massive protoplasm that moves and eats bacteria, and so forth. And of course, the lobate amoebae, you've already seen them in lab, amoeba, and you'll see them again tomorrow. And so, yeah, this is called dog vomit. It's not, this is a slime mold, okay? The fruiting bodies of a slime mold, those are the reproductive structures, and we're not going to go into the details of how they reproduce. But normally this particular group happen to be in individual amoeba cells that are going around in the soil individually. And when the food supply runs out, they all come together and they will have sort of like a mushroom. That's what it is up on top, and that's where they produce spores. It was interesting, several years ago, Mission School, not Mission School, but Mission San Jose School down here, just hopping a skip down the road here. They came down and or called me down there and asked, what animal is throwing up here? You know, it's all over the place. And turns out that it's this, just slime mold. Anyway, we won't pay any attention to them anymore. So the next group, oh yeah, apomorphy for this group. Is that the mitochondria? Do you remember mitochondria? What's the structure of mitochondria? There's the outer membrane and the inner membrane has broad folds in it, okay? These, the inner membrane, are tubes, okay? So they have tubular cristae, because those folds of the inner membrane, instead of being broad folds, they're bunches of little tubes, okay? So that's unique to them, and also, now most of these are unicellular, and they feed by broad pseudopodia, not the needle-like pseudopodia that we see in the Rhizaria, because they are also amoeboid. So that's an apomorphy. When I say lobed pseudopodia, we're talking about these broad, finger-like pseudopodia that instead of, in contrast to needle-shaped pseudopodia. Okay? And of course, this, they move by amoeboid motion. I strike this out, this is a mistake, because it isn't the movement of the cytoskeleton part is not the microtubules, they're microfilaments. Okay, these are microfilaments that form along the edge and sort of roll the membrane forward, okay? And that's what's happening here when you see the pseudopodia extending. It's really microfilaments, not microtubules. Okay, and we find them anywhere. Okay, freshwater, they're marine, they're really copious, especially the slime molds are in the soil. And usually you never see them. You never see them except for when they reproduce. Okay? And a few of these are parasitic. They cause diseases. For example, there's something called Entamoeba histolytica that causes dysentery, diarrhea. It's an amoeba. Sometimes it's in drinking water in certain countries that can really, in fact, it causes bloody diarrhea. It can really tear up your intestines. Okay, that's all. We're going to look at them, and the rest of it will piss the conduct. So this is the fungi and the animals. They belong to this group, and here the apomorphy is posterior flagellum when it's present, okay? Because there's some groups here, for example, a lot of the fungi have lost it. Everyone have this? So the fungi, okay, general attributes of these is that they're multicellular. So I'm putting this up as an apomorphy. Multicellularity has evolved independently in eukaryotes multiple times. So where else has it evolved multicellularity? In what we have covered already. Not Veridoplantae because there's chlorophytes that are unicellular. So the plants, that's one. Plantae, but Rhodophyta, independently. There are some chlorophytes that are multicellular. So it's evolved here 3 times.
There's also single-celled Rhodophyta, right?
There's what?
Isn't there also single-celled Rhodophyta?
Yeah.
So we can say that's an apomorphy, right?
The multicellularity, yeah, because they started out as unicellular. And then one other clade that is multicellular. It's not in that clade.
Amycota?
What?
Amycota? Amycota? The seaweed one? Oh wait, no, not Amycota. Phaeophyta.
Phaeophyta, yeah,
Phaeophyta,
okay,
yeah.
yeah, brown algae, that's right. And so this here is also twice formed. Once in the fungi and in the animals. Well, you say, wait a minute, let's go back here to that phylogeny. Yeah, why don't we put multicellularity here? Because there's things up here that I'm not showing that they are unicellular. Okay, so the multicellularity of animals and fungi are independently derived because there are unicellular critters down in this area. We're not looking at everyone, okay? We're just looking at the major groups here. All right, so some more general attributes. Multicellular, they have cell walls and of a structural carbohydrate called chitin. So does anyone remember what the monomer of chitin happens to be?
N-acetylglucosamine.
N-acetylglucosamine. okay? These are primitively heterotrophic. And in fact, this whole clade, Amorphia, is primitively heterotrophic. There is no evidence within this clade at all of having any ancestor that's photosynthetic. They're all heterotrophs. And this is by absorption. What it means is that the fungi grow through their food, and they secrete digestive enzymes outside of their body. These digestive enzymes digest the material around them into monomers, and then they absorb it in. Okay, so extracellular digestion outside of the body. And some of these are saprobes, which means that they're feeding only on dead material, dead organic material. And there's a lot of fungi that's exactly their food supply, is dead organic material. Tree branches, dead trees, leaf litter, and so forth. Some of them have become parasitic, okay, that they grow on living organisms. So, for example, jock itch in humans, aphix foot, those are fungi that are growing on your body. And some of them are mutualistic, that they're actually in a relationship with other species and they benefit each other. We'll come across all those a little bit later on. So the general structure of the body of a fungus is something, the whole body is called a mycelium. The whole body is a mycelium, and that mycelium consists of inner branching threads, okay, that are one cell layer thick, and they just branch all over the place. And each individual thread is called a hypha. And so the mycelium is the collective, all the hyphae that make up the body of the fungus. And usually you don't see the mycelium until it reproduces. When it reproduces, it has structures that we call sometimes mushrooms. Those are reproductive structures, but that is not the whole major body of the fungus. Fungus is usually growing in the soil or down in the substrate. Usually when you see it growing on your cheese at home, that green stuff that comes up there, well, the fungus has been there growing in the cheese for quite some time, and you just didn't see it. Because the green stuff that you're seeing are reproductive structures, and only then are you aware of it. So what is significant about this body form, all these little threads, is collectively all those threads have a large surface area. And when you consider how did they get their nutrition, by secreting enzymes outside of their body, digesting the material around them, and then reabsorbing it in. So large surface area is very important for rapid intake of material. If they had a solid body and only were absorbing on the outside of that solid body, then the amount of nutrition that they could take up from the surroundings would be very limited compared to the volume inside. Okay, so this is a form that creates large surface area. And some of these fungi can be huge. There is a mushroom, it produces mushrooms, Amilaria austrae. There was, I think this particular study was in Washington State in a forest. This is in association with the roots of a lot of trees, and they estimated that the actual fungus that's growing down in the soil, one individual covered 6.5 square kilometers. And so some of these fungi can be very, very big, and hence weigh, if you could collect it all, weigh several tons. All right, now this probably, well, I have a little bit here. The hyphae, they can be septate or they can be non-septate. Now what does that mean? So these hyphae are threads, and of course they have cytoplasm inside of it, Golgi, nuclei, etc. But it's easier to understand septate because the hypha is divided into individual cells. Each cell with a single nucleus, its own Golgi, etc. But the ones that are non-septate, they don't have that along the thread, because that thread is a tube. Okay, it's a tube structure. It doesn't have cross walls. So it's like this one right here. And so this is continuous cytoplasm. Nothing separates the nuclei. Now, the nuclei happen to be, spaced out about the appropriate place for one cell. And in the area of that appropriate space for one cell, there would be Golgi and so forth. But nonetheless, cytoplasm can move back and forth. And even with the ones that are septate, that there's usually a pore in that wall that still lets small materials move from one cell to the next. So this is non-septate. No, that's septate. This is septate. This is non-septate. This here is electron micrograph of septate. You can see that it's divided up into cells. And this is a major important thing. So those cells, those fungi that lack, that are non-septate, they're said to be multinucleate, or another word for multinucleate is cenocytic. Okay, cenocytic. All right, that's about probably all the further I can go. Yeah, let's just go ahead and stop here. Okay, slide 7. I'll briefly go over in lab life cycles, because a lot of the stuff you'll be looking at for the fungi will be having to do with life cycles, just like on the plants. So the plants material from yesterday, cross sections of lilies, those will still be in lab for those of you who are not finished with it. And that exercise 6 will be due at the end of lab tomorrow. We'll start exercise 7, but it will not be due. Okay, we'll put it off.